Good morning, good afternoon, good evening, everyone. Thank you very much for joining us for another episode of Emerge. I hope everyone is having a wonderful day. My name is Stoyan Stojcev, and I'm part of the Resin Biosciences team. Before we get into the webinar, the usual housekeeping announcements. We are recording the session, and we're going to post the recording in about a week's time on our YouTube channel, where you can find all other episodes of Emerge as well, in case you want to catch up with any of them. Please post your questions in a Q&A box. We'll have plenty of time to get to these after the presentation. And if you'd like to have your question answered live, please use the raise hand icon, and I'll activate your microphone after the presentation so we can have a little bit of a discussion. So before we get into the actual webinar and presentation, before I introduce Daniel, I just want to quickly divert your attention to this event. The Ubuntu Proteomics Summer School, which is going to run at the end of January 2023 in Stellenbosch, South Africa. The school is a week-long event, and it will be run by 14 facilitators from academia and industry. There will be parallel sessions, so we can cater for those just getting into proteomics or proteomics-focused projects with the beginner track, and for participants that are a little bit more experienced with the advanced track. There'll be plenty of time for discussions, networking. The people are encouraged to bring their own data to have project-specific discussions. So check out the website, ubuntuproteomics.co.za, and hopefully we can see you in South Africa. If not, please share across your network. It will be highly appreciated. So back to the matter of hand and the presentation today by Daniel. Who is currently a postdoctoral researcher at Novo Nordisk Foundation, a center for basic metabolic research at Copenhagen. So Daniel began his career into secretomics during his PhD, which he completed at the University of Guelph, Ontario, Canada, and where he discovered a novel stomach-derived secreted hunger hormone. And as I mentioned, he's currently working as a postdoctoral researcher in the Novo Nordisk Foundation Center for Basic Metabolic Research, where it utilizes the range of discovery mass spectrometry workflows in combination with in vivo and in vitro models of physiology to enhance understanding of health and disease. So today, Daniel will be telling us about some of the methods he's implemented in the lab for capture and processing of secreted proteins in high-throughput manner from a range of metabolic cells and tissues, and how some of these novel proteins play a role in the coordination of inter-organ crosstalk and energy metabolism. So, Daniel, first of all, thank you very much for joining us today. I appreciate the time that you've taken for this, and I really look forward to your presentation.
Perfect. Thanks, Stoyan, and thanks everyone for joining today. I'll just quickly share my screen. So you should be able to see.
Yeah, perfect.
Maybe get a cooler pointer here with the laser. Yep. All right. Yeah, thanks, Stoyan. And welcome, everyone. My name's Daniel. Don't let the mustache fool you. That is actually me in the photo. So today, I'm fortunate to be here on behalf of Novo Nordisk Foundation Center for Basic Metabolic Research. As Stoyan mentioned, I'm a postdoc there now. And I'm excited to talk to you about how we are using mass spectrometry to study secreted proteins in different metabolic cell types, and how we're using that to illuminate biology. So there we go. So just briefly as an overview, I'll talk about my research background. I know Stoyan already alluded to this, so I won't spend too much time. Some of the ongoing work we have at the Center for Basic Metabolic Research, or CBMR. How we sort of, I guess, come into dialogue with Resin Biosciences and using some of their bead-based preparations for secretomics, and then how, again, we're leveraging that to study secreted factors in health and disease, and in the context of exercise, and a whole bunch of different models. So I'll just quickly preface this by saying that I am a biologist by trade. So I'm sure there's many people in the audience that are far more technologically savvy than me. So by all means, challenge me on the technology, but I'm definitely a biologist by trade. So hopefully by the end of this talk, I can successfully merge the technology with how we're using it to study the biology. So very quickly, as you mentioned, I did my PhD in Canada. And here I was studying a hunger hormone called ghrelin, which is a stomach-derived hormone. And it really is what piqued my curiosity in secreted proteins in general. I just thought it was quite fascinating that you could have something that's predominantly secreted from one organ or one cell type that could then travel to distant organs and have metabolic repercussions. So it's quite profound in that sense. So it led me to pursue a postdoc at CBMR, and now I'm co-supervised by Julian Zirath, who's the executive director at CBMR, and Atul Deshmukh. So at CBMR, I'm a part of this functional proteomics and metabolism group. So as the name implies, we're trying to really marry the use of proteomics in studying metabolism. And I'm a part of the interorgans communications team. So for the focus of today's talk, I'll be talking about cell and tissue secretomics. I don't work quite as heavily on peptidomics and EVs, but those are also emerging areas in understanding interorgan communications. So for those of you that don't know, secretomics, it's simply the large-scale investigation or large-scale identification of secreted proteins. And as you can imagine, there's a number of approaches you can take to do this. And I'm just highlighting two, I guess, more contemporary approaches in the literature. So on the top, we have using microarray data or RNA sequencing data. And then you can look at how a gene might be differentially expressed in response to your perturbations. So we have, two studies here by Laurie Goodyear's group in Harvard in the US, where they exercise train animals, and then they take this RNA-seq data, and then they actually do bioinformatics filtering to identify potential secreted proteins. And then they go validate them as secreted proteins and study their effects. But what our group is more interested in is obviously using mass spectrometry to study things at the functional level or at the protein level. So we want to just sort of skip a few steps and jump right into identifying the protein itself. So on the bottom, we can see a few examples where we've used proteomics in media from cells to study the effects of, on the left, exercise, or in this case, you electrically pulse stimulate cells. And then on the right, a lipid-induced insulin resistance. And there you can identify novel secreted proteins that regulate physiology. So just to get everyone on the same page, I'm sure many people that attend these webinars are quite familiar with proteomics and the workflow. And this is something that we're actually using for review soon. So it's very focused on exercise and muscle. But the takeaway here is that the workflow doesn't really change much in terms of the basics. So you have a model and you have a sample type. So in our case, in secretomics, the sample type could be many things, but it might be media or conditioned media from a cell. You reduce the sample complexity by digesting the proteins, and then you load the peptides on the LC-MS. You identify them, you run bioinformatics, and you run stats and interpret your results. So the workflow itself is quite basic and quite similar to other avenues in proteomics. However, in secretomics, there are certainly some, I guess, unique challenges and unique considerations that I'll quickly elaborate on. And you might also wonder why we would even go into cells or tissue to study these things. And so the issue really is if you go into plasma is highly, there's a high dynamic range in plasma. And of course, anyone that does proteomics knows about the dynamic range issue. So you have these really, really high abundant proteins derived maybe from the liver or hemoglobins and things like that. And they essentially hinder your ability to identify not only low abundant proteins, but just identify peptides in general. So you get very low coverage, maybe in the 500 to 800 peptide identification range. And then cells and tissue are obviously easier to manipulate. You can give drugs, you can do knockdown experiments to knock down genes and proteins. And then the secreted proteins you're measuring are tissue specific. So that's also a benefit to that as opposed to looking at the plasma where there's a wide range of organs contributing to that response. So once you've selected your model and you've selected which somewhat dictates your sample type, you sort of have to use annotation databases in secretomics as well. So that's another unique thing with secretomics is we're always trying to add on layers to prevent false discoveries, I guess you could say. Since we know we're working with conditioned media and proteins that are in that media, but we don't really know if they're truly secreted or if they're an artifact of damage or something like that. So we use these annotation databases to filter for secreted proteins. But it's actually also interesting to note, before I forget, for anyone that does secretomics, you probably know that even when you annotate your proteins that you identified in the media of maybe your cells, your tissue, most things are still not annotated as secreted. So it's sort of a gap in our knowledge in the literature right now that we're trying to fill. So specific obstacles in cell and tissue secretomics are highlighted here. In the case of cells, there's obviously artifacts and serum starvation. So you need to wash out phenol and serum before you do proteomics on the media, because again, that would introduce the same dynamic range issue as plasma. So you could have artifacts of the physiology that are actually just due to removing serum as opposed to your treatment. It's also difficult to model chronic interventions in cells. So exercise and diet in particular, you can't really exercise train cells, obviously. And then the translatability is always a discussion point with cells. But then, so as you move towards tissue, it's great because it's a mature tissue. It has many different cell types, but unfortunately, those cell types also confound your ability to know where a secreted protein is coming from. It may not matter, but it may also matter to you. And then in some cases, as you'll see in today's talk, there's some tissue types like adipose tissue, in particular, when you do explants or adipose tissue explants, they're highly contaminated with lipids. So you really need to approach your sample preparation strategies to deal with each one of these obstacles as well. And then in the middle, we just have things that are common to both cell and tissue secretomics. So you have contamination from the buffer. The volume is typically very high, which is a bit annoying because it can be very laborious to concentrate samples. Although I'll show here in our workflows, you don't have to do that. The sample heterogeneity is huge. So depending on the cell type or the tissue type you have, you definitely need to factor that into your sample prep strategy, and then so on. So what really drew our attention towards using protein aggregation capture, or herein referred to as PAC, was this paper essentially pioneering this technology. And this is work from Jesper Olsen's group in the Center for Protein Research, which is also in Copenhagen. And the lead author was Tan Veer. And it's a really amazing technological paper that shows that you can use this technology in a wide range of sample prep workflows. And it can also operate in a wide range of starting material or starting input, which is really of interest for us since our group does tissue proteomics, expression proteomics, we do PTMs, we do secretomics, we do a bit of everything. So we found it quite interesting. The basis for this technology is that you can add organic solvents to your soluble proteins to create insoluble protein aggregates. Should use my cursor more here. And those can bind to the microparticles or magnetic hydroxyl beads, for example. And from there, you can wash your sample many, many times with harsh solvents. So you get a really pure sample, and then you can digest your sample on the bead, and then you can elute your peptides from the beads. So essentially, you'll get a very clean, very pure sample, and it sort of circumvents a lot of the challenges I mentioned just previously. The big thing with this too is that it's not only low cost, it's efficient, it's sensitive, but it can also be automated. And so that's something that we're also interested in, and I'll talk about in a second. So as we were reading this paper, fortunately, they also had some supplementary materials where they looked at the effects of different concentrations of beads on your, I guess, ability to capture or recover peptides from the beads. And we're curious to repurpose this application for secretomics. So we were really sort of, I guess, narrowing attention into the lower end here of samples that had lower concentrations or lower inputs. So if you see, as you start to get down to 0.03 or 0.04 microgram per microliter, and this is just a whole protein stain from the peptides that were eluted from the beads, you can see that you start to not really efficiently capture any peptides if you only use 40 micrograms of beads. So they just noted that you could actually just use more beads and you start to more effectively capture more peptides at the end of your sample prep. So that's something that we took into account when we were going to apply this to the Secretomics workflows. So that led over many, many months, or maybe even over a year, to the development of this workflow. And this is something that you can apply again to a variety of cell types, as I'll show in a second. And the basis of it is you have your model, and maybe you treat your cells or you treat your tissue, and you can collect your conditioned media from that sample. And then you collect it on ice, of course. You spin it at a relatively low speed, and then you can essentially remove some of the cellular debris and byproducts, damaged products. You can syringe filter it, again, just removing contaminants, apoptotic bodies, things we're not quite as interested in. And then it basically follows a regular proteomics workflow. So you denature your sample, we typically use urea, and then you alkylate it, reduce, and then you can go through with the pack sample preparation strategy, as I mentioned. So here we're using only about 5 to 10 microliters of this bead solution, which I think, if I recall, is about 100, 150 micrograms of beads. So it's quite a low amount, and each sample would receive that essentially. And then you can desalt your peptides as usual and then run it on LCMS-MS. So this is the basic skeleton of the workflow we're doing. And as I mentioned, the benefit to protein aggregation capture is that you can also upscale this process. And so we're very fortunate at CBMR to have this Kingfisher Flex purification system. And this is one of our PhD students, Roger, gazing lovingly at our robot. And we love the robot because it allows us essentially to load all of our plates with our samples, with our digestion buffer and our washes. And then we can load those washes onto the robot. Press start, it'll run through the protocol overnight as we sleep. So it's very nice and allows you to do, I guess, 96 samples in one run. So it's very nice. So I mentioned a major consideration when you're doing secretomic sample preps, at least in metabolic cell types. That's what I know the best, I guess, and what I've been working on the most. A major consideration is the heterogeneity of these cell types. So it wouldn't be surprising to think that different tissues or different cells can have a different level of secretory capacity. And I think that's sort of what we're seeing here to some extent. I tried to simplify this graph, but there's a lot you could take away from it. But in the basic sense, we have here 2 cell models. WT1s are also a cell model. And then in the middle here, we have skeletal muscle types, soleus and EDL. And then we have inguinal white adipose tissue. So these are mature tissues. And in one case, we took the whole fat pad, and in one case, we actually minced the fat pad. And then we took the media and we ran the sample prep. And I think the interesting thing here is that if you, for example, compare adipocytes here, so C3H10s to C2C12s, which are myotubes or muscle cells, it looks like they have the same starting protein concentration, but it's actually not the case because I prepared the C2C12 media twice and then I pooled the peptides. So it wasn't until I did that we got a comparable level of peptide. So on one hand, it could mean that this model just secretes fewer proteins, but it could also mean you have different starting number of cells, which is also a consideration. But nevertheless, it's something you certainly have to work out a bit in your lab, depending on the metabolic cell type you're working with. But if we move towards the tissue here, this is soleus muscle. It's an oxidative muscle. And this is EDL, which is a predominantly glycolytic muscle, and that's referring to their fiber type distributions. And soleus, you can see that there's far more peptides in the media after the sample preparation. So I think that to some level could be just because it's a more oxygenated tissue and it has maybe a bit more serum or blood contamination when you take it out of the animal, even though we do wash it. Or it could suggest that there's major fiber type differences between these 2 muscles in terms of what they secrete. And then interestingly, if you look at the WT1 cells, for example, these are brown adipocytes from mouse. We can't detect anything, actually, and I've done that a few times and I still can't detect much. So maybe a different sample prep strategy has to be used here. Or it's possible you, again, need to stimulate them in some way, but it's just more an observation than anything, and it's a consideration for your own research endeavors. So now that I think I've sufficiently harped on the technology, I can start to move towards how we're using the technology to study the biology. And in the first avenue, as I mentioned, I'll be looking at cell and tissue secretomics. So the first avenue is cell secretomics. And the basis for this study was actually quite simple. It was just trying to identify novel regulatory myokines, or muscle-derived secreted proteins, that are secreted from muscle in response to an oversupply of glucose. So of course, that could mean maybe you're consuming a high carbohydrate meal, or it could mean that you're diabetic and you have a high level of glucose just in your blood at rest that your muscle is seeing. It's surprisingly a question that hasn't really been addressed in the omics area of the literature. So to do this study, we decided to fully differentiate C2C12 myotubes, as a model you saw in the previous slide. And these are contractile myotubes, so they're sort of a mature myotube, although there's still a cell in the dish. We treated them for 16 hours with either high glucose or very high glucose, we'll call it, 40 millimolar, or a control. And the control for these cells is actually already a bit high glucose because they don't differentiate otherwise. So its control is osmolarity matched 25 millimolar glucose plus 15 millimolar mannitol. So quite simple experimental setup. You can see, well, the image is a bit blurry, but you can see that the cells look relatively happy at the end of that treatment. So there's no issue probably with the viability of these cells. So once they've been treated, we wash them 4 to 5 times, and then we leave on a serum and phenol-free media, and then we collect it after 4 hours. And that's our 4-hour conditioned media that we can proceed with sample prep. So from there, as I mentioned, we do urea denaturation, we do the semi-automated pack prep. And as I mentioned earlier, we do 2 times preparation for C2C12 because that seems to be what works. And we use 250 microliters of unconcentrated media. So it's purely just taking that syringe filtered and spun media. So we do that twice, and then we pull the peptides at the end of the desalting, so at the end of stage tipping and everything. And that's what we run on the LCM-SMS. So here, I don't talk a lot about instrumentation, but we are also fortunate to have these instruments at our disposal. And so I think a lot of the data you see is actually owing in part to the state-of-the-art instruments. But we ran it on an NOLC with a 60-minute gradient. And then this was run into the timsTOF Pro 2 mass spectrometer. And it was operating in DDA mode. So from there, we identified almost 2,600 proteins, which was quite good. And we were quite happy with that. And then with a fairly strict cutoff of a minimum of 3 identifications per group, we quantified just over 900 proteins in the conditioned media. Among those proteins, 180 were annotated as, I guess, highly confident secreted proteins. And I've just put above there what I use to filter for secreted proteins, although many people do this quite differently. So either looking at a uniprot signal annotation, which means the proteins have a signal peptide sequence, so they're probably being trafficked through the classical ER-Golgi pathway for secretion, or they have a secreted annotation, meaning they've been validated, which is usually very few of them. But I also look at proteins that are annotated to the extracellular compartment. So 180 secreted proteins in this experiment, and most of them were actually differentially regulated with a high glucose. So within the secretome there, so among these proteins in the media, this is just noting that they annotate to many different cellular compartments, so not just extracellularly. They're also annotated intracellularly in the nucleus, in the mitochondrion. So it's just suggesting that secreted proteins probably don't just have a role outside the cell, probably also have a role in the cell. And there's definitely a growing body of evidence that suggests that the localization of these proteins is really what dictates their function. So it's termed moonlighting, I guess, so they can have different effects depending where they are. But nevertheless, We get nice separation of the groups here. So we can see the control here. There's a little bit of variability, but the glucose, the high glucose group clusters quite nicely. And there's most of the variability attributing to component 1 here. And then if we look at the PCA loading analysis with the secreted proteins highlighted in purple, I guess, not pink, purple, you can see that a lot of these secreted proteins are sort of what's driving that separation among the group. So that's good. That's what we sort of were expecting to see in the conditioned media. If we look at a volcano plot here, you can just tell that high glucose is potently regulating the secretome. So this is now looking not just at secreted proteins, it's the entire conditioned media. I'm showing this because our main goal with this study was actually to identify and shortlist candidates that had regulatory effects in metabolism. And we don't really want to miss something just because we didn't annotate it as secreted. So, you know, if we see in the media here that something has a really big fold change or it's highly significant, but it wasn't annotated as secreted, we might still be interested in it. So that's sort of why I'm showing this here. But the really simple takeaway here is that, again, there's potent regulation of the muscle cell secretome by just high glucose exposure. And then these secreted proteins are implicated. Some of these are already well known as being implicated in metabolism, like adiponectin, which is more classically known as an adipokine, but also myokine. Insulin growth factor binding proteins. You also get a lot of proteins involved in ECM remodeling, like CRIP2, MMPs, or matrix metalloproteinases, as well as stress responses, so heat shock proteins. So understandably so, since I'm sure this is a bit stressful for the cell. So we were then trying to take it a step further and really hone in on some candidate regulatory proteins that had functional effects on metabolism. And so this is just, you know, a snapshot of some of the candidates that we found interesting from the screen. I'm really honing in on this S100X protein. That's not its full name, but that's the name I've given it for this talk. And I think these S100 proteins were quite interesting because you only see them identified in the high glucose group. So I thought they could be particularly interesting in terms of what they're subsequently doing in the muscle cell and maybe even in distant organs. So when I just did a simple ELISA for this S100 protein in lean, obese, and diabetic plasma. These are humans or human plasma. And it was interesting to just note that it was actually significantly reduced in the diabetic plasma. So completely opposite to what we might expect after identifying this in a high glucose screen. So it's just worth noting again that plasma is a complex sample. Many organs are contributing to this response or this observation. So there's definitely a lot to unpack there that we don't really know yet why. But nevertheless, we decided to pursue whether this S100 protein had any direct metabolic effects. Before we did that, we of course needed to decide what was important to check. And for those of you that maybe aren't biologists or not as well versed in the metabolic field, I'll just briefly mention in obesity and diabetes, in skeletal muscle, glucose uptake can be impaired, at least insulin-stimulated glucose uptake. So we were curious whether this factor could be regulating glucose uptake in any way in the muscle and whether it worked with insulin in any way. Maybe it could help insulin's effects. And then in the liver, we know with sort of a chronic progression in diabetes, at least maybe later on in diabetes, you can have ectopic lipid deposition. So a lot of this oversupply of lipids, either from the diet or from a dysregulation in their mobilization from your fat stores, they can progress towards being deposited ectopically or in other organs like the liver. And so again, we were also wondering if this protein we identified could be somehow regulating the handling of lipids in the liver. So that was sort of the basis for it. And now we'll get to briefly some of the metabolic effects we've seen. So we got this recombinant form of this S100X protein. And first we looked at its local effects in the muscle cells. So this is the same myotube model that we identified it in. And we saw that, so this is the control bar, and this is glucose uptake. We just took an area under the curve of glucose uptake in the cell. So insulin stimulates glucose uptake here. And we can see that this S100 protein, even without insulin, has a similar effect to insulin, which is really interesting. And then when you give this protein with insulin, there's no potentiation of its effect. So it seems that this S100X protein increases glucose uptake in an insulin-independent manner, which is very interesting because, again, in diabetic muscle, there might be some level of insulin resistance. So we're always trying to find factors that can work independently of insulin. When I check some of so these are Western blot data, when I check some of the classical signaling markers that could regulate glucose uptake, so AKT and TBC1D4, we didn't see effects, but we did see effects when we gave the recombinant protein in pink here on CAMK2 activation. So it suggests that maybe this protein is working through that signaling pathway. We then again went to look at the liver, so if it could have maybe endocrine metabolic effects, so endocrine simply referring to the fact that we identified this in a muscle cell screen. And we see that at least at a 1 micromolar concentration of this recombinant protein, we see a reduction in fat oxidation in these human hepatocytes when compared to the control. So it suggests that this S100X protein acutely attenuates palmitate oxidation in human hepatocytes, at least in vitro. So it sort of jives with the idea that, you know, if you're having these chronic oversupplies of glucose, this protein might be secreted and migrate to the liver to have potentially damaging effects over the chronic or the long term in diabetes. But again, there's a big gap in our knowledge that right there. So we have to certainly pursue a lot more. So this is sort of our working model. I mean, I just sort of emphasized it, but we have a high glucose exposure, which gets taken up and in some way regulates the myotube secretome. So we get a secretion of many different factors, the differential expression of many different factors, one of which is S100, and it has autocrine effects on stimulated glucose uptake in the muscle, but it might also crosstalk with the liver where it can actually attenuate lipid utilization in that cell. So just briefly, I will go over our second avenue, which is looking at tissue secretomics. This study is much more, I guess, preliminary, we'll say. So I just thought it might be interesting to include because, again, it's highlighting that you can use pack sample preparation even on a mature tissue in secretomics, which really not many people have done to date in the literature. So the goal of this study was to identify, this time, adipose tissue-derived secreted proteins, particularly in the context of exercise. And you might wonder again why we're trying to move towards tissue. And again, it's just because the translatability might be a bit higher. This is just one example I thought was worth including because it's quite a straightforward and simple example to highlight the fact that even leptin, which is a very well-known adipokine, it's well characterized in terms of stimulating satiety. If you look at these C3H10 stem cells, which get differentiated into adipocytes, you can see that you can't actually detect leptin even with a commercial ELISA. So you really start to question this model as in terms of its ability to recapitulate what's actually happening in a mature tissue or in vivo. Whereas if you take explant, so inguinal white adipose tissue or epididymal white adipose tissue, which is more of a visceral adipose tissue, you can easily detect this adipokine. So really, we're always trying to move towards tissue. And until using this sample prep strategy, it was difficult to do because of the lipid contamination. So why adipokines, you might also wonder. And it's really just because they've been well documented as being potent regulators of metabolism, either in the context of exercise or even as you progress into obesity. The adipose tissue remodels, and it seems to really impact how these tissues secrete adipokines to regulate metabolism and insulin sensitivity. So they're certainly a hot topic. So as I mentioned, it's a very challenging sample type to work with mature tissue for secretomics. I did an initial test that, I mean, I didn't even want to dignify these data by showing it, so I just put it in text. I used an in-solution digestion here. For those of you that know, you just essentially prepare the sample and digest it all in a single tube. So there's no transference. So you definitely get a good yield of peptides, but you get a very contaminated, very dirty sample. So we identified almost 1,000 proteins, but you can tell already that's not great. And we had way too many missing values. So there's clearly a reproducibility issue there. We had poor digestion efficiency and a lot of missed cleavages. So again, we were really excited to try this protein aggregation capture on this sort of model. So what we did, and I'm close to wrapping up now, what we did here is we exercised animals for 1 hour. We anesthetized them and perfused them in the heart with PBS. So that's sort of our idea here, was to clean all of the organs and rid them of serum and phenol contamination, or not phenol, but serum and blood contamination. Then we excise the tissue and we wash it in a large volume to clean it. And then we again collect the conditioned media. So again, this media is very contaminated with salts, buffer salts, lipids. So we use a protein aggregation capture sample prep. And so what we found here is that we could identify almost 3,500 proteins in that conditioned media. So very, very promising. And with a minimum of 5 identifications in at least one of the groups, we quantify almost 2,500 proteins. So this experiment I didn't mention, but we had 3 different adipose tissue types. So we identify a lot of secreted proteins, actually. We get almost 721 secreted proteins. And as I mentioned, it's very preliminary, so I can't really talk too much further about it. But all I can say is that we are seeing at least major differences across the different types of adipose tissue depots, which is unsurprising since they have very different types of metabolism or a metabolic phenotype. So it wouldn't be surprising if that extends to their secretome. So if we just look at the differential expression of the secreted proteins, so this is only secreted proteins in a visceral on the left versus a subcutaneous white adipose tissue, we can see that there's massive regulation. So we identify low abundant proteins like IL-6, but also immune function related proteins like CD14, again, ECM remodeling proteins like MMPs. So it's just showing that, again, this is definitely a viable approach even for mature tissues ex vivo. So in conclusion, I went a little bit over time, so I apologize, but well, for my own timing. This sample preparation in general remains a key consideration in proteomics, and I'm sure everyone is quite aware of that, but I'm just emphasizing that since we've had a lot of obstacles along this way. Protein aggregation capture is definitely a viable approach for high throughput secretomic sample prep. And you can use this and apply this to a variety of different metabolic cell types. And I hope we've evidenced that today a little bit. And then PAC is particularly useful when you have heavily contaminated samples. So in our case, when you incubate cells or tissue, you have these high salts, high lipids. So it seems to really, really work well for that. And again, I can't really talk about secretomics without talking with the instrumentation. And when you couple the PAC prep with these instrumentation, you can certainly detect a lot of low abundant secreted proteins that probably have important metabolic roles. So we're excited to continue this, and we'll see what we can uncover in terms of biology moving forward. But with that, I'd like to thank everyone for listening. And of course, I'd be happy to answer any questions. And thanks again, Rezan, for having me.
Hi, Daniel. Thank you very much for the presentation and letting us know about the work you're doing in the lab. It is very interesting to understand some of the workflows you've developed. So maybe I'll kick off with one question, a little bit more technical focus, I suppose. So you highlight the relationship between protein concentration and the ability to capture protein using this method. And then you have different types of cell types and tissue types that you see that they secrete different amounts of proteins. So do you then match the amount of beads to the amount of secreted protein based on the tissue type, knowing upfront more or less how much is expected, or how do you go about that? So do you do some kind of concentration determination upfront for each sample?
Yeah, so it's a good question. I think we've talked about this a lot in the lab, and ideally we could do some sort of protein assay to begin with in the starting material. So if you had a conditioned media from muscle versus adipose versus whatever, you could then really hone in on exactly the optimal ratio of beads. But because you can't really do that to our knowledge in any clean, simple way, we rely on doing it after the fact. And we haven't repeated a lot of these experiments since I've tested them. So I think now that we have the knowledge, of approximately what starting material we have in each one of these cell types, I think we can more accurately couple the beads to the protein, or at least get the optimal ratio for sure. And I think it's clear that some of them are warranting further tests, like these brown adipocytes I've just struggled with. So maybe we again have to use more beads, and then maybe prep them a few more times and cool the peptides. You know, it's just a bit laborious that way, but yeah, I think we're getting there.
Yeah, definitely. So maybe a follow-up question on the front end is, I noticed that you're using urea for the actual denaturation. It's quite interesting because typically SDS is used, but I see that potentially there's some link between which is the best denaturant and concentration, because we had some very recent observations that with urine, which is also quite a dilute sample source of proteins, actually urea works better with some of these bead-based methods than SDS. So I was wondering if you've actually tried different denaturants up front, and how did you arrive to urea?
Yeah, I'm sure someone before my time tried SDS. We certainly use SDS pretty much exclusively when we work with tissues. But yeah, you're right. With conditioned media, I think I don't really have a good answer, because I think initially we were doing pack manually, and one of the issues when you do it manually with SDS is if you have any left over, it can be an issue later on, essentially. So
Sure.
the robot, I think, has circumvented that. So I think, to get to your question, we haven't really tested it, but it's something we could revisit now probably, because we have the automated preparation and not so worried about leftover SDS in the tubes. So yeah, I'm actually not sure. And I haven't also played too much with varying the concentration of the urea. I believe it's 2 molar in the final
Yep.
concentration. But yeah, it seems to work well. So it's good to hear that you also had a similar experience with these dilute samples. Yeah,
that makes sense. So we got a couple of questions coming in. So 2 questions from Lenspers. Hello, Len. She's asking how much media you begin with, and the follow-on question, what type of filter do you use to filter the media?
Yeah, so the starting media can, I mean, I suppose it depends. It depends on your plate format that if you're talking about cells, for example, it'll really depend on that. Like if you have a 12 well plate, you might have 1 ml of media, and then I'll just take an aliquot from that, and that seems to work fine. But we're also trying things all the time to reduce the amount of media we have during the 4-hour exposure to collect those, basically to get more peptides in a more concentrated sample. But yeah, I've worked from anywhere from 1 ml of starting media to even 2 mLs in tissue as well as cells, and I just take 250 microliters from that, and it still works. Yeah, but again, there's like infinite combinations you can do on these things. And then, sorry, the second question was the syringe filters.
What type of filter do you use for the media, to filter the media?
Yeah, it's just a 0.2 micrometer cell strainer or cell filter. Very basic. It's a syringe filter essentially. So you attach it onto a plastic syringe, and then you load your media, and you carefully syringe filter it through. I haven't tried not doing that. It just makes me feel like I get a more pure sample, but we're not actually sure completely.
We have a question from Sudeep. Have you tried Rapidgest in your sample prep? I suppose more on the solution, but you could probably add Rapidgest also on the on-bead method as well.
To answer it briefly, no, we haven't tried that actually at all.
What is your digestion efficiency look like typically with the pack methods and the Secretome analysis? Do you have some kind of ballpark figures?
Yeah, again, we're not too sure because we don't know our starting material. We're kind of working backwards to calculate what we have at the start. But it seems typically with pack, we lose maybe half. So 50%.
I think with dilute samples, this is probably expected. Yeah.
I think it's expected, yeah.
So I have maybe 2 questions, maybe one again for the front end, experimental design more. Do you see the need to do quite more replicates when it comes to tissues because there's probability of having larger biological and technical variability. Typically, what do you aim for in terms of when you do the tissue-based secretomics?
Yeah, it's a really good point. I would completely agree. In the past, we were aiming for 4 to 6 probably. But I think ideally, and this is sort of holding true even with a lot of tissue proteomics now too, probably 10 to 12 seems to be more robust. Of course, you should always aim higher. But it's the same with cells. We're trying to aim for higher number of biological replicates, and then you sort of can, you can have multiple plates as a technical replicate, and you can pool them, and then you can have you essentially reduce the variability that way. And we've done a run recently with that, and it's worked very, very well, and the clustering is really, really good. So yeah, I think with tissues, it's especially complicated because when you take the tissue out, we're only as good as our hands allow us to be to dissect, and sometimes you might be getting maybe a different lobe of the adipose tissue. Maybe you didn't quite get it all. Maybe you got some other contamination. So yeah, I think with especially tissues like adipose, which have a lot of cell types, you need to do like 8 to 10, 8 to 12 probably. Yeah.
Okay. We have another question that came in. Do you think centrifugation could work to remove cells and debris instead of filtering, or do you think this would spin out the secreted vesicles?
Yeah, it's a good point. I think that's why we've tried to keep the spin really low, because I know you can use, differential centrifugation to isolate, EVs and other microparticles. I would just basically err on the side of using a lower spin, but we do lose peptides. We know that when we do the spin, you're essentially losing at almost every step of the process. So it's a bit tricky to know. I have never tried actually just not spinning it yet. So I could definitely try that and see. Yeah.
I think from a little bit of experience we have in our lab, if you keep the centrifugation low, below like 1000 Gs, it should be okay. And I think vesicles start spinning out more at higher speeds. But yeah, something to check, maybe a non-centrifugal centrifuge sample.
Yeah, I agree.
So to jump into the back end of the workflow, and you mentioned the importance of the way the data is searched in terms of the databases. So can you elaborate a little bit on this? You don't use basically a full Homo sapiens database. You would do a Secretome specific database. You generate maybe added contaminants to it. Is this the approach?
Yeah, do you mean for annotating something as secreted, or?
Yeah, but even so, your initial spectral matching, I suppose, do you do that with a full Homo sapiens database, or you still have a sub database with just secretory proteins potentially?
No, yeah, that's a good question. No, so for the spectral matching and the computational, the quantification is you're still matching it against the in silico digested mouse or human FASTA. Okay. Yeah, so it's still, that's for the quantification, it's still that. And then, for the annotating after that, you would, you can do a lot of things. There's these third-party databases, Vesiclepedia, Exocarta, all these other ways you can try and find maybe non-classically secreted proteins. But they're a bit more contaminated, right? So depends on the purpose of your study. So we have one published study last year where we use those databases, and of course it boosts the number of secreted proteins you have, but you're not really sure whether they are truly there. But for this study, I was more focused on, again, identifying something that's confident and pursuing its effects. So we didn't use those third-party ones. Yeah.
Cool, fantastic. So I don't see any more questions coming in. If there's anything last minute, just let us know. You can drop another question. But meanwhile, this really awesome presentation. Thanks again for coming on board. We'll do another episode. And since we don't have any more questions, I want to thank you and thank everyone for joining and taking the time for this webinar. And we'll see you guys for the next one.
Thanks for having me, Stoyan.
Cheers, Daniel. Cheers, everyone.
Cheers.
Bye-bye.